already met some of you, but I'm Jillian Rowe, and I work for the New York Abu Dhabi Center for Genomics and Systems Biology. I work in the bioinformatics core, but it's an entire center. Yeah, so today I'm going to talk about the bioinformatics core sequencing, our general infrastructure, give a little bit about Anaconda and Conda, which I guess everybody already heard a little bit about, but I can go more into it, and the Gen Core modules, which obviously we deploy with EasyBuild. Yeah, so this is kind of an overview of what we do. Up here we have the core sequencing. So we do our own sequencing in-house. We don't always send them off to Illumina, but I think we might have Illumina machines. I'm not sure. So then there's also a lot of other things that go in here. We have QC. We have whether or not it passes or fails QC. Things fail QC a lot. The analysis of all of this kind of data. Over here we have all of our own workflows. We have multiple databases that we maintain, things that are publicly available from BLAST and CBI, all of these kind of things. The stuff over here is mostly what I'm going to be talking about today, that we have the Gen Core HPC modules, which are deployed with EasyBuild. And down here, we have all the other kind of things you would expect: logging, visualization, analysis, interpret results, and scientific project management. We are also starting to do training and trying to have more of like an online presence with resources, documentation, that kind of thing. We kind of like to dream big in Abu Dhabi, and we'd like to have at least a semi-automated infrastructure, all the way from building software to deploying them on the cloud. And bioinformatics is a little bit different in this kind of case for some reasons that I'm going to get into. All right, so then, if you want to see that over here, this is basically what I just talked about. We build the software packages. We build and test software stacks. Software stacks are done functionally. So for instance, they're grouped by analysis type. So these would be things like variant detection, RNA-seq, microarray, if that's still around, metagenomics, this kind of thing. So the point is that you should load a module for the type of analysis that you're doing. You shouldn't load it by tools. We upload the software stacks to the cloud, and we deploy the software stacks on Delmo, which is our HPC. Over here, we have the actual analysis, because this is right, like our end goal. We have lots and lots of data that we produce, but at some point, we want to be able to tell a story with this data. So on this side, we have create composable analysis workflows, compose and evaluate HPC directives. The idea behind this is that we work quite closely with New York. We use Slurm, they use PBS. From our point of view, they're almost the same thing, right? So there shouldn't be a huge hassle between using the two. Run workflows on the HPC, and again, create public resources. So we do quite a bit of internal development and testing. Like we've set up the infrastructure. We have, for the most part, this is where I tend to spend a lot of my time, is my users request software, and I do my best to install it. We have some of our own software that's built in-house, and of course, this is probably the bulk of the work right here, is the scientific development of the actual analyses. These take quite a while because you can go through and read a paper, and of course, things do not always work exactly as the papers describe, and so then there can be quite a bit that goes on here. Yeah, so this is the goal. This is what we want to get to. These are all pieces of software that come up in an analysis, and this should be one module in an ideal situation. It's not always an ideal situation, but this is a variant detection workflow. What you do is you have, where is it? Right up here, you have a bunch of sequencing data that you align. So usually what happens is you have like a reference genome, and then you're doing a bunch of like string matching with some kind of parameters against the reference genome to try to find what are called scaffolds. And so you're just aligning your DNA against a reference. And there are a few different kinds of software that do this. You do quite a bit on like just converting between file types, cleaning sort of that initial stage, sorting it. You do quite a bit of post-processing on those alignment files. You can do this step over here, which is relating to reference data sets, which is a little bit of what I talked about before. Like we supply all of these data sets and try to kind of streamline this process for users a little bit. Down here, you we do what's called variant detection. So when you have DNA, you know, for the most part, we're all basically the same, but there are these changes, and the easiest ones are called single nucleotide polymorphisms. So it's one change in a DNA, and that one change can do, you know, it can do nothing or it can do something. And that's quite a bit of the idea sometimes, is to find that out. So all of these steps are kind of to that purpose. Yeah, so then you can see, I mean, each one of these, so this is 1, 2, 3, 4, 5, 6, 7, 8. Well, this here is like, so this brings us up to at least 10. So just as like a first pass, this is 10 different pieces of software that we're using. And this doesn't include everything that these softwares depend on. So now I'm going to change gears a little bit and talk about Anaconda. We already sort of know what this is. It's just a scientific distribution of Python. I like it. It is much easier to install on an HPC system than compiling Python from source. And it has the Conda package management system, which I really, really like. And I'm going to go more into that. Another sort of favorite aspect of this is the Anaconda client, which is just an API that gives you information about your packages. Okay, back to bioinformatics workflows. This is a bioinformatics workflow. It is duct tape upon quite possibly more duct tape. And sometimes we like hit it with a hammer and try to get it to work. Yeah, and this is because, you know, most of the bioinformatics people that write software, they have a very specific knowledge about a very particular, like, biological problem. And they can solve that biological problem, but it might not necessarily use, like, software best practices. So it works sometimes, and then it's our job to make it try to work in a more, like, robust fashion. And of course, we want to be able to put all of these, like, little answers and solution to problems. We want to be able to put those together so that you can have, you know, this nice, big, pretty analysis that we showed here. And, you know, another goal that we have is that the software environment should hopefully remain the same throughout the entire analysis. So, you know, I should not be using some version of Picard here and another version over here, right? They should always be the same. So if you left users kind of to their own devices, it would be really, really easy to make that mistake. Yep. Yeah, so, you know, most bioinformatics teams are kind of small, so when possible, we try to collaborate. You know, the biggest ones for us are, where are they? Conda and Conda Env. Conda Env is now a part of Conda Core, but it's, Conda Envs are basically like, they're like meta packages. So you just say, I want all of these softwares, and I want to group them all together, instead of building, like, you know, instead of saying individual software packages or saying Conda install some big line of things. Bioconda is like, you know, they're my favorite software group, I think, of all time, because they already have so many bioinformatics softwares built, and that means that I don't have to build them, and that makes me pretty happy. They do have some problems, like, for instance, their build process has really matured over the last, you know, like 2 or 3 years, and so sometimes you still have some legacy packages in there that people don't use, or they haven't been rebuilt for whatever reason, and then, you know, those can cause problems. But for the most part, it's really good, it's really robust, it's perfect for HPC because it uses the Conda package manager, which should hopefully assume that you don't have any system dependencies. And then, of course, EasyBuild is what we use to deploy our modules. Yeah, okay, so this is this here, can you guys see that? Because I know the black background is kind of horrible. All right, it's just like a list of software. What is this from? Yeah, this is from variant detection of the workflow that I showed earlier. So you can see there's a lot of software in here. If you were going to do, you know, module load for this analysis, you would be doing a lot of module loads, and that's actually exactly what we used to have. Like, I used to go through and help people set up aliases in their bash RC to do, you know, basically all of this to make sure that they had their environments right per project. And obviously, this is a much nicer way to do it. Yeah, so I think all in all, this is about 250 like individual software packages. Oh, I just want to take a minute to mention the channels up here. Each channel is just a different repository of Conda packages, and usually they're done you know, by group, and each group has like a specific interest. So Bioconda contributes bioinformatics software. R contributes packages for the R language. The NYUAD CGSB, that's us. Basically, I just have like meta packages and documentation packages up there. Module load GenCore variant detection version 1.0. And then it gives you everything that you need. Yeah. So everything is tested in the cloud using Travis. And I just wanted to show that quickly if I can find my mouse. I was saying earlier, like every time I do a presentation, I really have problems finding my mouse. Did I find it? Did I find it?
It was there.
It was there? No, this is going to give me like anxiety, you guys. Where's my mouse? Oh, oh. Where is it? Okay. Yeah, see? Oh, this is Bioconda, and look at that. That's like so tiny. Oh, I don't know. I mean, if you're interested, also Bioconda builds using Travis as well. So a lot of my emotional state for any given day is how many of these green check marks I have, and I get really upset when they're all red.
What are you testing exactly here? Just the installations or also?
Just the installations, yeah. You can't often, I'm going to get into that later, you can't often test like real world cases because it's building in the cloud, and so you have the problems of like how do I get, you know, the necessary data there, and how do I get real world data there, and the build is only like, I don't know what, so long. I think it's like 2 hours or something.
2 hours to build the whole kit?
No, the maximum build in Travis I think is around 2 hours. So you couldn't do like an entire, you couldn't do an entire analysis probably. Conda Forge is another channel that I really like. They have more like general use software. They have stuff like CMake. I forget what the other ones are, but CMake was the one that I used from there. And I think they're starting to have like R packages and things in there as well. I was going to try to find the GenCore ones. Oh, now see I have all these Conda Forge ones in here. What is this? Okay, sorry, I'll try to, I have a URL for it someplace. But because they are Conda environments, they all get uploaded to the cloud, so you can also see them on, you know, Anaconda Cloud, which is quite nice. So another reason for this was because, you know, any of our users anywhere, even if they're not on an HPC system, they're not necessarily using modules, they can still go and get the software environments from Anaconda. So we've tested this with a few different like lab servers, because it's quite common that a lot of the groups will have their own server, and then they sort of go and try to do things on their server for a little while, and then eventually they get tired of that and come to HPC. But in the transition step, they do have all the environments. I'll find the URL soon. Haha, here they are. Okay. Yeah, I mean, it's a pretty standard Git workflow every time somebody requests an environment, or a module rather. So we do project specific, and we do like the GenCore kind of production modules. You know, it gets built. Hopefully there's the green check mark. When there's the green check mark, it gets merged. It gets uploaded to the cloud. We all rejoice until it stops working. How do I make this go back to full screen? Oh, there we go. Yeah, I already talked about Bioconda. I don't know. I think the best thing about it is just that like it has a really big community support. So usually if there's something wrong with the package, if it's being used, you know, someone will eventually come and report an issue. There's also like a few different people on the team who go through and, you know, kind of do some quality check of like this package is okay and this package is not okay. Just to kind of illustrate that point, I want to show the Anaconda API. So I can do Anaconda show, Bioconda, SAM tools. This is the version. This is the build. It's being built for Linux. There's also the options to build for Windows and Mac. Bioconda does not build for Windows, but some of the other channels do. And then it's SAM tools. This is the version. Okay, and then this is the build. So software can be rebuilt, usually because there's a problem with it, but there could be other reasons. Okay, so down here I have the dependencies, and you see the first time around that this was built with no dependencies. And this is wrong. There should be dependencies. So I think it was that there was a problem with some of the dependent packages, and so they just built it like this, because not all of SAMtools requires those particular packages. But then you can go and look at the 0.1 release, and you can see that then there is ncurses in zlib, which is the way that SAMtools should be. Yeah? So I really, really like this to just check out packages and see if there have been any changes or what the changes are. And if I want to, I could probably go and really investigate those on GitHub. We have just a very small application for the most part. It's just hooking into the Conda code, building the environment, making sure that none of the packages conflict with each other, because you have that quite often with bioinformatics software, and then uploading them to the cloud, and then it also creates the EasyBuild config. It all started because I raised an issue on Git. The specific reason is because Anaconda will choke and fail if it has the LD library path set to the Anaconda prefix lib. So you can't set that, and it's because Anaconda is using R paths. I don't actually fully understand why, but the point is that it will choke and die. And so initially I had to build an easy block that did not set the LD library path. And so I went online, and Kenneth helped me, and then he asked me if I would contribute it back, which I did, and it got quite a bit of cleanup. And now there's a Conda easy block. You can see these are the Gen core modules that we have deployed. I think there's actually quite a few more now, because we also have project-specific ones. So if there's kind of a long-running research project, for instance, if somebody wants to write a paper on it, I will build their environment, I test it, I upload it, and it lives there forever, and then they can use it. So, I mean, one example is, it's not here, but for this RNA-seq, there are a bunch of RNA-seq projects, and they all have slightly different requirements because they were started at different times, or they're doing slightly different, post-analysis and this kind of thing. So in terms of future work, I want to add a few more options to the Conda easy block. I need to add a lot more checks with EasyBuild using real data. This is something that I got into because just because it builds does not mean that it works. And that happens a lot. So we want to set up a service, using EasyBlock somewhere where it's testing against real data. And we have this for most of our analyses now. Open collaborations and, create infrastructure for modules as a service. We're already kind of most of the way there. You can always go and just grab the environments from Anaconda Cloud, even if you're not using modules. I don't know. I have a little thing up here about collaborate. We should all collaborate because, we just should. We have lots of publicly available resources. We're trying to have some more. Oh, actually, since it came up before I stopped, I just wanted to bring one thing up because the chyme example came up a few times. And this is what the chyme recipe looks like. So you can see it's a very difficult recipe, even for Bioconda. You hardly ever see this kind of stuff here, where they have very specific version requirements. Matplotlib tends to be, they break their APIs with every single solitary release. So that's one you tend to have to be very, very specific. Yeah, they do. Most releases anyways. Burrito filling, I don't even know what this is, but it just gives me a lot of problems. Yeah, I don't know. I forget what this means. I think it means skip everything except for Python 2.7. Maybe somebody who uses, is it chyme or chyme?
Yeah, I see the chyme not the movie before. But this might be the format not the movie before. Maybe. Probably can. Oh, except this doesn't have a fixed version.
No, you can have fixed version. See, this is a fixed version.
Yeah. But what this is talking about is also generating these config files from this. We want fixed versions and just the versions. Yeah, but it's
Well, you can kind of have that, right? Because this one is like greater than, equal to, not this one.
This is YAML, right?
This is YAML. Yeah, and the parser already exists in Conda. I tend to steal a lot of code from Conda. They have some good stuff in there. I mean, it's the same kind of thing, right? You have your build requirements and your runtime requirements. You have some tests down here about... Huh? No.
They have the whole list duplicated. This is just asking for values. I mean, they have matplotlib both and build.
But you can use... I mean, you can use variables, right? It's something like...
Yeah. So magic.
Okay. Yeah, no, there definitely is. Because I know the Conda Forge is much more strict about that. Like, they have a linting service and all this kind of thing.
It's also built included. Like EasyBuild. I don't see why.
Sometimes.
This is just asking for properties. Sooner or later, you're going to get a mismatch between the versions. Anyway.
That is a good point. I'll bring that up. Well, you don't always want to require a specific version, right? I don't know. So far, I like it. I suppose I never thought of the fact that you're basically just duplicating the list most of the time.
Something to try hard to avoid an input, having duplications. Because it's going to stab you in the back.
Yeah. I mean, if you wanted to see quickly how Conda builds, they build against a Docker image. And the Docker image has some kind of magic on it so that it has like no system dependencies. But it tells you like all your neat stuff. There's the channels here. This is kind of irritating to me about Conda. If you don't have your channel priorities right, you will break everything and like absolutely everything. So this is a good way to go through and check and make sure that your channel priorities are correct. So you can see here that it just, it just gets all the packages that you need and tells you some other things. It runs the tests. This part down here is kind of important because you can see that it's running tests. And it creates like a tar for you that you can then upload to Anaconda Cloud. Yeah. And I think that's it. Acknowledgements.
You want to pass around the mic or questions? Or?
There are questions.
Yeah, I have one.
Well, didn't answer the mic when you asked the first question.
First question.
No questions for you.
Second question is, can I mine? So one thing that comes into mind is you seem to be using Conda quite heavily. Are you concerned at all about the builds that you use being generic builds, especially on the HPC system? I've seen the advantage of using Anaconda because you can let your users just deploy them on their Mac and play around. They can get familiar with the environment and then they switch to the HPC and it's exactly the same. So they don't run into surprises. But you're using generic builds everywhere. Have you ever looked at how big of an impact that has in terms of performance?
No, because most of our software isn't going to be optimized to use like anything kind of special. Like if you try to use the Intel compiler or something with it. I mean, so many of these things don't even make on the first pass anyways that I wouldn't even try to optimize them.
Yeah.
So for us, it's not a concern. It would be nice if someday it became a concern.
Yeah.
But for right now, it's just not.
I mean, extension of that, are you using R from BioConda? Or from Conda?
From R, yeah. From the R channel of Conda.
And that's not configured with SIMD stuff. It's all OptArch generic then.
Yeah.
Because we saw on our bulldozer AMD like a 4 times speed up for our programs just for the naive, silly things that people do locally. Like they would, and whenever they're mapping something, they'll get basically the speed up, the SIMD type speed up. So I would suggest comparing, say, EasyBuild R to your Conda R and see if you get a difference, because even with GCC, we found like you'll get the AVX compile and you'll be able to test it anyway.
Okay. No more questions. We'll move on, I think. I don't know how we are for time, but I feel like we're falling back. So.
Okay, thanks guys.